Hi, I work on brain organoids, and today I'm going to tell you about some engineering approaches that we're taking to organoids in my labs. Brain organoids are self-organizing 3D models of the human brain that we grow from stem cells, and they develop in similar ways to the brain in a human embryo. People are really excited about these as a model system. They let you study a lot of things not possible in other traditional systems, but they have quite a lot of variability, and that limits experimental reproducibility and the conclusions that can be drawn from them. This is the standard procedure for generating midbrain organoids. So after forming cell aggregates, we embed them in a hydrogel scaffold to support 3D growth. And we think one of the major sources of variability is the hydrogel that we use, called matrigel, which is an ECM extract from mouse tumors, and it's extremely variable batch to batch. So we ultimately want to replace matrigel with a synthetic hydrogel, with the idea that this would reduce organoid variability and provide a better, more reproducible model system. So that's the big picture goal. And the first thing we're investigating, and the question I'm focusing on today, is how do the mechanical properties of the supporting hydrogel affect organoid growth and development? So we're using alginate to do this, another hydrogel material. And we're mixing alginate with matrigel to create composite hydrogels that we can modulate the mechanical properties of. So we can tune the stiffness by adding more or less alginate, because stiffness is known to be really important in tissue engineering and can affect cell proliferation and cell fate. We want to keep matrigel in there for now because it has lots of growth factors and cell adhesion motifs that are important for organoid growth. So adding alginate to matrigel lets us study the question about stiffness without changing these other parameters. So my lab mate has done some mechanical characterization of our composite hydrogels, and this shows you that we can indeed modulate the mechanical properties of the composite by adding alginate. So we can think of storage modulus like stiffness. So the more alginate in the composite, the stiffer it is. So the alginate 1% composite is our stiffest gel. Now, once I started growing organoids in the different gels, the very first thing I did was a live dead stain. So 100% matrigel is the standard for growing brain organoids, but you can see really good viability in all conditions, validating further work with our composites. And then here you can see some images of the organoids growing over time. So in the stiffest gel, you'll notice that the organoids are much smaller than in the other conditions, and this is quantified here. In all the other gels, the organoid growth rate is very similar, whereas in the stiffest condition, growth is restricted. So the stiffness of the hydrogel the organoid is embedded in affects its growth rate and final size. So then we wanted to know if the stiffness makes a difference to what's happening on the inside of the organoid. So here are some stained tissue sections, and the bright sort of squiggly actin staining in green highlights structures called neural rosettes. They are characteristic developmental structures in brain organoids. These are where you can really see the self-organization that happens in these organoids, and all of the development and differentiation is centered around these structures. So what you'll notice is that there are many more of these structures in the 100% matrigel condition than in the stiffest hydrogel condition. And the rosettes are also bigger in the 100% matrigel. And this is quantified here, where you can see much fewer rosettes on average in the stiffest condition. And so then I measured the size of the rosettes by tracing them. And you can see from these histograms that all the rosettes in the stiffest gel were very small, while the other three conditions had a much wider range of sizes with much bigger rosettes. So what we think this means is that the stiffness of the gel the organoid grows in affects its ability to form these rosette structures, affects its self-organizing capacity. So here's some other staining that I did for neuronal markers MAP2 and tyrosine hydroxylase TH. And you can see that we have neurons in all conditions. So even though rosette formation is affected, we're still getting differentiation and maturation into neurons. We're working on quantifying this right now, but by eye, it actually looks like we have more neurons in the stiffest condition. So maybe stiffness is also affecting differentiation and maturation of progenitor cells into neurons. The main conclusion that I have from this work so far is that the ability of brain organoids to self-organize depends on the mechanical properties of the material that they grow in. So it's critical to take that into account going forwards. whether for engineering a new or synthetic material or for trying to model processes of brain development or disease. Thank you.